My name is Amanda Vozella, and this is the Gut Microbiome Project. The gut microbiome is defined as a proportion of microorganisms in a given environment. In this case, the gut is the environment. Based on the gut-brain connection theory, the gut microbiome affects both mental and physical health. A poor microbiome can influence IBS, colon issues, and various mental illnesses such as depression. The microbiome is most at risk for deteriorating at infancy and old age. It is important to keep a good proportion of microorganisms at these stages. The studies where we did IBS, Andrew Schreiner researched and showed that the gut microbiome is correlated to diet, environment, and several aspects of health, including obesity, diabetes, and irritable bowel syndrome. His scientists and him discovered that microbes found in the gut contribute to metabolic functions, protect against pathogens, and strengthen the immune system. So those with a weakened immune system can get IBS. And also depression. Zhang and his team discovered with bacterial evidence that the gut microbiome affects mental health, especially depression. The proportions of actinobacteria and bacteroides alter the amount of serotonin and may influence a person's likeliness to develop depression. Our question was, how does age affect the gut microbiome? For our hypothesis, we said that as age increases, the gut microbiome becomes more stable and there's less risk for poor gut health. However, when the organism goes into the end stages of life, it deteriorates again. Our rationale for our hypothesis is that as infants, our gut microbiome is only developing, so it's at risk for disease. Meanwhile, when we grow older into the end stages of life, the body begins to deteriorate, and so will the proportion of the microorganisms. We did, for animal 1, we did 307, who's Hoshi, a dog, a flat-coated retriever who is 6 months old, weighs 50 pounds, is female, has not sexually matured, and it's fed Mother Hubbard biscuits, table scraps, yogurt versus dry food and grass. And this is the niece to our other animal, 308. So animal 2, 308, was Nova, also a dog, flat-coated retriever, 4.5 years old, weighs 50 pounds, female, also fed Mother Hubbard biscuits, table scraps, yogurt versus dry food and grass, and is the aunt to animal 307. For week 1, we gathered DNA from the fecal samples in order to use the Quagan kit and Zymo kit to isolate and concentrate the DNA. The Quagan kit is specifically designed for the isolation of DNA from fecal samples. To do this, we added a buffer solution to prevent the DNA from degrading and to bind the DNA to the filter within the small column. We washed away impurities by using a series of washes, wash buffer AW1 and wash buffer AW2. Then we transferred buffer ATE into the column to disrupt the bonding between the column and DNA. To guarantee DNA isolation, we used the following solutions: inhibit ES buffer, buffer AL, lysate, wash buffer AW2, proteinase K, ethanol 96, 100%, wash buffer AW1, buffer ATE. The Zymo kit is used to concentrate the DNA, 10 times the initial amount of DNA. The DNA from the Quagan kit is bound to a filter in the Zymo column, rinsed of any impurities, and eluted in a special buffer at 1/10 the volume of the initial. We use the following solutions to guarantee the concentration of DNA increase: CHIP DNA binding buffer, DNA wash buffer, and DNA elution buffer. For week 2, we isolated and concentrated DNA from fecal samples from specific animals The specific gene we wanted to analyze was the 16S gene, which allows us to determine which types of bacteria were present in the animal. It has 9 variable regions and is frequently used to determine classifications such as the genus of an organism. First, we determined the concentration of the DNA, and then we amplified the 16S gene using PCR, specifically the B3 and B4 regions of the 16S gene. To determine the concentration of the DNA, we used UV spectrophotometry because DNA absorbs very well at the wavelength of 260 nanometers. After determining the concentration of DNA at the 260-280 nanometer ratio, determine the purity of the DNA. If the concentration was between 1.5 and 2.2, if it was less than 1.5, it was probably contaminated by proteins, and if it was greater than 2.2, it could have been contaminated by RNA. We added a series of solutions which added the necessary components for PCR to occur: primers, nucleotides, and DNA polymerase. The primers had to bind to the targeted sequence of DNA before the DNA polymerase could bind and begin adding nucleotides to the 3 end of the primer. To begin the PCR process, the temperature of the DNA must be raised to 95 degrees Celsius. After the primers bound, the temperature of the solution was raised to 72 degrees Celsius so DNA polymerase could bind and begin synthesizing. To make the PCR reactions, the solutions were put in a thermocycler. Each cycle of PCR potentially doubles the number of the target molecules. For week 3, we checked to make sure the PCR reactions from week 2 were successfully using gel electrophoresis. Electrophoresis relies on the charged particles in a solution to migrate in an electric field. The DNA migrated from the negatively charged electrode anode towards the positively charged electrode cathode. DNA molecules have a negative charge because of the phosphate groups in the backbone, which causes the DNA to migrate towards the anode. We can calculate the size of the DNA fragments by the distance the DNA molecule travels and is inversely proportional to the logarithm if its molecular weight. The expected size of the amplicon was to be about 550 BP. After the PCR, we had to clean it up by removing the components of the amplification PCR buffer, which is Mg2+, DNTPs, and enzymes. Then any unused forward and reverse primers. The Egencourt AM per PCR cleanup system theory is the double-stranded DNA will be bound to solid magnetic beads. The single-stranded DNA with the primers will be washed away with solutions and other impurities. Then we determined the nucleotide sequence of the fecal samples in a large mixture containing all of the other student samples. We had to mark our specific samples by adding the last 2 digits of our animal-specific number. We ran the index PCR for 8 cycles since we tried to add a short sequence, and instead of making a large number of amplicon. For week 4, during the beginning of week 4, all of our previous data was lost and we had to use the past semester's data to finish. The Egencourt AM per XP kit is used to remove the components of the amplification PCR buffers and leave the DNA, which was binded to a magnetic surface, to minimize the amount of DNA that was lost. We used a fluorometric assay to determine the concentration of the amplified sample after the impurities were removed. We did not use UV spectrophotometry because it cannot determine the difference between proteins, RNA, and DNA, and would result in a higher concentration value than what it truly was. In contrast, the fluorometric assay can target a specific nucleic acid and give accurate concentration values. During week 5, the data was received, which contained the results of the gut microbiome's diversity at the following levels: kingdom, order, and genus. For UV absorbance spectrophotometry, spirometry results. We had the older dog, number 308. The concentrations were 29.7 nanograms per microliter, and the 260-280 ratio was 1.67. And for the younger dog, the concentration was 15.8, and the 260-280 ratio was 1.84. The DNA ratio should be approximately 1.8, which indicates the DNA concentration is good. 260-280 ratios of 1.5 to 2.2 are considered to be within the acceptable range, and both of the dogs were found to be within this range. Using the concentrations determined from spectrophotometry, we calculated the amount of DNA that will be put into the PCR reactions. For our number and quality of reads for each sample, 07E307 had total reads of 2,687, and it also had 2,687 reads passing the quality filter, so its percent reads passing quality filtering was 100%. For 07E308, the total reads was 18,028, and that's passing quality filtering was also 100%. The positive 3AP had 2015 total reads, and that was also 100%. And our negative control 3DN had 98 total reads, and the percent reads passing was also 100%. This chart identifies the total amount of bacteria reads for each sample and the positive and negative controls. It also shows the quality of the reads that pass through the sequencing. For gel electrophoresis was done to check the products of the PCR reactions. The control shows where the band should be found if the PCR reaction was performed correctly to isolate the 16S ribosomal gene. The negative control showed DNA present, which indicates that there was contamination, and the dog DNA did not show up, so that means that there was not enough DNA present to make a band. And then for our kingdom level, we have this bar graph, and the graph shows the percentage of classified bacteria shown up in the reads at the kingdom level for each sample. It also shows the percentage of unclassified bacteria. For classified, the older dog showed 99.02%, and the younger dog showed 75.25%. And for unclassified, the older dog showed 0.98%, and the younger dog showed 24.75%. And then we did the order comparisons, and this got more specific from the kingdom level. So this graph shows the vast proportions of different bacteria in the orders. The green bar is for the younger dog, while the blue bar is for the older dog. And then we also did the genus comparisons, which is more specific than the order. So that graph also shows the different proportions between the bacteria genus, and the green bars for the younger dog, the blue bar for the older dog. And then we have the tables for percentages for the order and the genus. And that shows the percentages and proportions of each bacterium in the order samples and the bacterium genus samples. And then for our discussion, the DNA extracted from the stool samples was successful. For each sample, we removed 500 microliters of isolated DNA, After using UV absorbent spectrophotometry, we determined that the DNA was pure enough for each sample. For the DNA, the 260-280 ratio should be approximately 1.8, which indicates the DNA concentration. The 260-280 ratios of 1.5 to 2.2 are considered to be within the acceptable range. The older dog had a ratio of 1.67 and the younger dog had a ratio of 1.84, meaning both of the samples were found to be within the range. The gel electrophoresis was done to check the products of the PCR reactions. The control shows where the band should be found if the PCR reaction was performed correctly to isolate the 16S ribosomal gene. The negative control showed DNA present, which indicates there was contamination. And the dog DNA didn't show up, so that means there was not enough DNA present to make a band. During the start of week 4 that our data for the original sample was lost, samples were lost, so we were unable to determine if there was a high enough concentration after PCR cleanup for sequencing using fluorimetry. The positive control had way over 1,000 reads that passed the quality filter, and 100% of those reads passed the quality filter. At the kingdom level, 99.02% of the older dog was bacterial, while only 0.98% was unclassified. This means that the positive control had little to no contaminants, since it's expected to be a high percent of bacteria. Since the positive control is E. coli, there was a small amount of cross-contamination. The gut microbiomes of the 2 animals we chose for our project had similar kingdom level because any animal on this has the same bacterium. As we get to order, it becomes more specific to the species. Finally, the genus level becomes vastly more specific to the species. The percentages became more specific as we went down from order to genus. This supports our hypothesis that the older dog would have more diversity in the gut microbiome than the younger dog. We found that at the genus level, the older dog had 6 different genus classifications greater than 6%, while the younger dog had 5 that were greater than 6%. At the order level, the older dog had 4 order classifications greater than 6%, and the younger dog also had 4 classifications greater than 6%, but they were lower percentages.